Hello, my name is David Rusch, and this presentation is about identification of major root diseases in BC.
This slide was adapted from the BC Ministry of Forests publication, Managing Root Diseases in BC. It lists the major root diseases and provides their scientific names and ranges. Armillaria, black stain, and laminated root disease occur only in the southern half of the province. Tomentosus occurs throughout BC, but is more common in northern BC. A more complete description of hazard by root disease can be found in the BC Ministry of Forests buyback portal. This table was also adapted from the BC Ministry of Forests Managing Root Diseases in BC, and it lists the susceptible species for all the major root diseases in British Columbia. The laminated root disease is primarily associated with Douglas fir, and tomentosus root disease is primarily associated with spruce. There are two subspecies of black stain. One occurs on pines and the other on Douglas fir. And armillaria root disease has a very wide host range. It affects all native tree species in British Columbia. All root diseases cause similar types of symptoms in their host trees. This particular picture is a Douglas fir tree. And you can see reduced height growth. That's usually the first symptom. Sparse chlorotic or yellow foliage, and if you look really closely, you can see very small, numerous stress cones. These are smaller than normal cones that are formed just before the tree dies, and then the final symptom is death. If you compare the tree on the left, the healthy tree on the left, you can compare the height of the leader with the tree on the right that has a very short leader. Two of the root diseases, armillaria and laminated, form root disease centers. And when we do a pre-harvest assessment for root disease, we're actually focused on mapping these centers rather than individual trees. Centers are formed by the root-to-root spread of the fungi and are characterized by openings with snags and symptomatic trees around the edges and blowdown in the center. Larger centers often fill in with less susceptible species, depending on the particular ecosystem. This slide shows a laminated root rot center, and this is looking up in the middle of the center, and you can see it's fairly open, and then around the outer edge we can see snags and symptomatic trees with typical root disease symptoms. This photo shows a laminated root rot center on the coast. You note the snags, and it's fairly open, and western red cedar is a less susceptible species for laminated root rot, is coming up in the middle of the center. This slide shows a laminated center in the interior Douglas fir zone. In this case, the center is regenerating back to Douglas fir, but you'll note the regenerating Douglas fir is quite a bit younger than the trees surrounding the root disease center. Also note the snags and symptomatic trees around the edges of the center. With armillaria root disease, the centers are often less well defined. This picture shows a large armillaria center in the interior of BC. There are a number of less susceptible deciduous trees scattered together with healthy-looking Douglas fir and the occasional Douglas fir snag. In these instances, the centers are defined more by the timber type rather than as discrete openings. This photo is taken near barrier in the spring, and in this instance, the deciduous tree patches indicate areas of root disease, and the surrounding Douglas fir areas are areas with little or no root disease. This is a large 4-hectare laminated root rot center in the southern interior, and the open canopy and difference in species composition allows these centers to be seen in aerial photos. This photo is from the coast, and typically on the coast, centers are smaller than they are than some of the centers in the interior, and it's difficult to map or see individual root disease centers. In this case, I cluster a group of smaller centers has been mapped as a single stratum. In this particular photo, root disease centers have been overlaid on an aerial photo. This is from the coast, and you can see it's not actually possible to delineate the root rot centers from the air photo, but you can. There's some evidence of root disease by the open nature of the areas where the root disease is present. So we talked a little bit about symptoms that root disease cause in their host trees, and root rot centers that are formed by armillaria and laminated root rot. Now we're going to move to talking about signs of the different root diseases. I'm going to start with laminated root disease, and this slide shows 3 photos of laminar decay. On the far left, the wood is actually delaminated. As the laminated root rot, it's easier for the fungus to digest the spring wood than the late wood. But you'll notice in the other 2 pictures you don't get that delamination. So it doesn't always happen that you get that clear delamination. The decay is characterized by very small little pits, and these brown tannish areas are actually areas of concentrations of setal hyphae, which I'm going to talk about on the next slide. This is a close-up picture showing the setal hyphae that I talked about in the previous slide. These are characteristic of laminated root rot. They're larger than normal darkly pigmented hyphae, and when you look at them under hand lens, they look like little brown whiskers. And they often form, or they're often associated with zone line formation, and they're not always present in the decay, so it just depends on the particular decay sample you're looking at. Laminated root disease spreads by ectotrophic mycelium. That is, fungi that grows on the outer surface of the root. You usually have to go down to the mineral soil to see it because the organic layers have a lot of competitive microorganisms that inhibit the spread of the fungus. In this picture, the soil is being removed from the root, and you can see the white mycelium spreading along the outer surface. Most wood decay fungi form dark zone lines, black lines in the decayed wood, and these serve to protect or keep out other decay fungi and microorganisms. Laminated root rot, though, often forms these zone lines on the exterior of the ectotrophic mycelium, or in this case, the root's broken off and it's formed this exterior zone line at the break, and that's this brown, crusty looking layer. Eventually, this will turn dark black. When you disrupt it with your fingernail, it often makes like a crinkly noise. You can also see in this picture, because the fungus is spreading along the outside of the root, it kind of acts as a glue, and you can see the rocks and dirt are stuck to the outer surface of this exposed root. The advanced decay of laminated root rot is preceded by a brownish purplish color stain. In this picture, you can see it on the butt ends of the logs in a log deck. The stain breaks down quickly when exposed to light, so you have to look just after harvest in order to see the stain. This is a picture of a laminated root disease fruiting body. It's just made up of a pore layer. We call these resupinate, and they're not particularly useful for identification because they only they're really hard to find. They're usually found on the undersurface of logs, and so you have to turn the logs over to see them. This one is a fresh example. They're only alive for 1 year, and then they turn a dark brown color after that 1 year period. I'm going to move on to talk about armillaria root disease now. This is a picture of the honey mushrooms, the typical mushrooms that armillaria forms in clusters. They usually form at the base of the infected tree in the late fall, before the first frosts. So they can be very useful in identifying the root disease if you just happen to be there at exactly the right time. In addition to typical symptoms of root disease, armillaria also causes basal resinosis or sap flow at the base of trees in many cases, and you can see that in this photo. The resin concentrate at the very base of the tree. Sometimes you'll see resin on trees from wounds that occur higher up in the tree, but then it runs down. But in this case, the resinosis is just associated with the base of the tree, and there's no indication of any injury higher up in the tree. The easiest way to identify armillaria is by these mycelial fans that form underneath the bark. This is a piece of bark that's been flipped over, and you can see the characteristic fans growing under the bark in the cambial layer. This is the easiest way to identify armillaria root disease. When we're just talking about armillaria root disease in the context of forest management, we're usually talking about Armillaria stoia, which is a species of armillaria that actively kills conifers. There's also a species of Armillaria sinapina that's mostly saprophytic on conifers. In the previous slide, I showed you the typical fans formed by armillaria on live host material. Once the tree's dead, it typically you just get sheets of mycelium growing under the bark. And when this happens, it's very difficult to distinguish Armillaria sinapina from a stoia. The general rule of thumb is if you're seeing it killing dominant and codominant trees and you get those nice characteristic fans, it's likely Armillaria stoia. If you're only seeing it on dead roots or on really suppressed understory trees, it's quite likely it's Armillaria sinapina. One way you can distinguish between the two species of armillaria is by their rhizomorphs. These are rhizomorphs of Armillaria sinapina. They're commonly referred to as shoestrings, and they can spread from infected roots through the soil in search of roots of susceptible trees. Initially, after clear-cut harvesting, a lot of infections occur through these shoestrings. But as you get root-to-root contacts becoming more common, then this becomes the main method of spread. This shows the difference between the branching pattern of the shoestrings of Armillaria sinapina on the right and Armillaria stoia on the left. Armillaria sinapina has monopodial branching pattern, whereas Armillaria stoia has a dichotomous branching pattern. If you look at a dead deciduous tree and remove the bark, you'll often see extensive rhizomorph networks of Armillaria sinapina under the bark. It's typically a lot harder to find the Armillaria stoia rhizomorphs. You have to look carefully on exposed roots to see them, and they're easily confused with fine roots. The decay of Armillaria just looks like water-soaked wood. So it's not particularly diagnostic. One interesting thing, though, about this fungus is the decayed wood actually bioluminesces. So you can see this if you look at the decayed wood at nighttime. The fruiting bodies don't bioluminesce, just the decayed wood. Next, I'm going to talk about a black stain root disease. This particular fungus actually doesn't decay the wood. It's a vascular wilt. It's vectored by insects, secondary insects and weevils. And black stain's one of the most difficult diseases to diagnose because you have to actually cut into the wood to see the black stain, and it only usually disappears after a couple of years. So you have to look at a recently dead material to see the stain, this characteristic stain in the wood. The last root disease I'm going to talk about today is tomentosus. And it has this characteristic honeycomb pattern of decay when you look at the ends of broken off roots. And this is one of the easiest ways to identify it, is to break off pieces of root on blowdown and look for this typical honeycomb pattern. When you look at the decay from the side, you'll notice it has these very, very long pits that are visible in the advanced decay. The early stages of tomentosus decay are very difficult to distinguish from decay caused by Phalaenus pini. You get these cellulose-filled pits in both cases. The picture on the right shows the fruiting body of Phalaenus pini. It's a true heart rot, and that's associated with the branch stubs. Sometimes you'll get both of these occurring on the same tree, so it can be a challenge to distinguish which one is present, but typically tomentosus will be only found in the roots, whereas the Phalaenus pini would be in the main bole and not extend very far down into the roots. The advanced decay of tomentosus is preceded by a brown stain, in this case in the heartwood. Tomentosus forms these very characteristic fruiting bodies. On the left you can see one's been flipped over, and on the right you can see what it looks like from the top. These are sometimes confused with hydnum species, but the hydnum species actually have little spikes rather than pores on the undersurface like tomentosus does. And again, you have to be there at the right time, usually in the fall, or you're not going to see these fruiting bodies. The easiest way to survey for tomentosus root disease is right after harvesting through a stump top survey. It is possible to do a pre-harvest assessment with using a drill or increment bore. And this particular, on the photo on the left, you can see drilled chips. The top left is healthy chips, bottom left is stain chips, and the bottom right is chips with advanced tomentosus decay. And you can also see in the increment core, you can also see the advanced decay in this particular core. If you're doing a post-harvest stump top survey, you just look at the freshly cut stump surfaces for stain or honeycomb decay. In this photo, you can clearly see the honeycomb pitting. In this case, the tree was cut with a chainsaw. When it's cut using a mechanical harvester, it's much more difficult to see the honeycomb pitting. This slide is just a review showing the different signs of the different root diseases. It also gives the 3-letter codes used by the BC Ministry of Forests. So the D stands for disease, the R stands for root, and then the last letter distinguishes which type of root disease we're talking about. So this is particularly important if you're doing a free growing survey in British Columbia. When you trying to identify root disease in young stands, I follow this basic procedure. So I'm looking for clumps or individual dead symptomatic trees with typical root disease symptoms. And then I will look, if I'm there in the fall, I'll look for armillaria tomentosus fruiting bodies around the base of the tree. But at other times of the year, I will expose the root down to mineral soil and look for, on the surface of the root, I'm looking for evidence of ectotrophic or mycelium caused by laminated root rot. If I don't see that, then my next step is to remove the bark and look under the bark for signs of armillaria fans. And if still nothing comes up, then my last step would be to cut into the sapwood and look for evidence of black stain or tomentosus staining.
In mature stands, it's a lot easier to identify armillaria and laminated root rot because we just look for the root disease centers. And to identify the type of root disease, we use the kick or chop method. Look for a blowdown, chop into a root, and look for characteristic decay or evidence of armillaria fans. For tomentosus, you likely won't see individual centers, but by looking at all the spruce blowdown in your stand for evidence of the honeycomb and decay, you can determine if tomentosus is present or not. If there's no blowdown present, then you have to use the same system that we used for young stands, what I'm calling the LERC method. Look for fruiting bodies, expose a root down in mineral soil and look for ectotrophic mycelium, remove the bark and look for fans of armillaria, and then finally cut into the wood looking for black stain. That's the end of this presentation, and thanks a lot for listening.